Hello, everyone, and welcome to this presentation, which is part of the AnaBios webinar series. I'm Andrea Ghetti, and I'm the CEO of AnaBios Corporation. AnaBios is a unique contract research organization based in San Diego, California, entirely focused on translational research conducted utilizing human tissues and organs that are recovered from ethically consented donors. We have developed methods and compositions that allow us to maintain viability and functions of the recovered human tissues and cells. And these technologies enable the study of drug activity in human ex vivo preparations or the investigation of pathological mechanism of disease states in human tissue. Over the years, we have applied these methodologies to support translational research in a variety of therapeutic areas. And we have been especially active in pain research, which with work conducted on human dorsal root ganglion neurons. But more recently, we have expanded these activities by developing tools and capabilities to recover viable samples of the human spinal cord. In a major effort, which involves collaborations between AnaBios, Eli Lilly, and the groups of Mike Hildebrand at Carleton University in Canada and Mike Iadarola at the NIH, we are now exploring novel approaches to study human spinal cord circuits using a combination of advanced transcriptomics methods and functional live tissue imaging. Today, we will have two great speakers that will provide a glimpse of what is possible with these novel approaches. Our first speaker will be Mike Iadarola. Dr. Iadarola is currently a senior research scientist in the Department of Perioperative Medicine Clinical Center at the NIH. He received his doctoral training in pharmacology at Georgetown University Medical School, where for his thesis, he investigated anti-epileptic agents modulating GABA receptors. Dr. Iadarola was then a postdoc at the Duke VA Neurology Research Laboratory and later at the National Institute of Mental Health under the direction of Drs. Erminio Costa and Xiu-Yin Yang. There, he identified the fundamental role for the endogenous opioid peptide dynorphin in spinal cord-related mechanisms of pain. He further pursued this finding after a move to the neurobiology and anesthesiology branch of the National Institute of Dental and Craniofacial Research. And in 2014, Dr. Iadarola received the Frederick Kerr Basic Science Research Award from the American Pain Association. This is an award that recognizes individual excellence and achievements in pain scholarship and is presented to pain professionals whose total career achievements have made outstanding contributions to the field of pain research. As it will be evident from his presentation today, the overarching goal of Dr. Iadarola research is to generate strong basic science from which we can achieve tangible therapeutic advances. His laboratory is currently conducting major investigations using RNA-seq to elucidate the primary afferent and spinal cord neuronal transcriptomes in mouse, rat, dog, and human to explore the full transcriptomic response in inflammatory states and in nerve injury, and create a new pain nociceptome that could be used to identify new analgesic targets. Our second speaker will be Dr. Vivekanand Jeevakumar. Dr. Jeevakumar is currently a scientist at AnaBios. He received his PhD in cognition and neuroscience from the University of Texas at Dallas. And during his PhD, he worked on the role of NMDA receptors in schizophrenia and showed that early postnatal ketamine administration in mice results in long-lasting cognitive deficits caused by abnormalities in inhibitory synapses. He did his first postdoc in the Department of Psychiatry and Behavioral Neurobiology at the University of Alabama at Birmingham, where he studied the contribution of calcium channels to neuronal network synchrony in the cortex, and later moved to the pain neurobiology research group of Drs. Ted Price and Greg Dussor at the University of Texas at Dallas, to study the mechanisms underlying chronic pain and migraine. And here he used patch clamp electrophysiology to show that interleukin-6 upregulates the calcium channels CAV3.2 in mouse DRG neurons in a mean K-dependent mechanism. At AnaBios, Dr. Jeevakumar leads the development of human spinal cord live imaging assays. Each presentation will last about 20 minutes, and we will leave time at the end for a Q&A session. Please feel free to use the chat box at the bottom of the webinar interface to enter your questions. And also in the handout sections, feel free to download the AnaBios spinal cord brochure. All right, Dr. Iadarola, please take it from here.
Thank you very much, Andre. And thank you for the kind introduction and the invitation to speak here. Today, I'm going to talk about 3 topics: transcriptomics, immunocytochemistry and in situ hybridization, and single nucleus sequencing in the human spinal cord. So there are no conflicts of interest, and the views presented do not reflect those of the US government. The main question I'd like to address is, how do we use this to further our understanding of pain and analgesic mechanisms? So with transcriptomics, in other words, sequencing every mRNA that's in a particular tissue, all of the data that I'm going to show is derived from RNA sequencing. And I will show one application to gene discovery and species validation, namely itch, gastrin-releasing peptide, and PPB, another neuropeptide, natriuretic peptide B. Immunocytochemistry and in situ hybridization will look at neuropeptides and nociceptive and analgesic receptors or ion channels. And then with single nucleus sequence, we extend this to the single cell level and look at nociceptive and analgesic receptors with a focus on endogenous opioid systems. And these are just examples of what can be done. Obviously, with an enormous amount of data at your fingertips, you can ask any question that you want or explore any finding that has ever been reported in the literature. Now, to look at transcriptomics, let's quickly take a look at one of the most fundamental questions that one might want to ask before or immediately after making a particular finding, and that is the gene expressed in the target tissue? So this tends to validate the original hypothesis. Is it in the cell type and the circuit of interest? And to what degree and how much is it expressed? So this latter question can be answered with sequencing. And the question that we'd like to ultimately look at is, can we devise metrics to answer how much is enough? How much expression is enough to make a molecule worthwhile for investigation? I actually don't have a complete unequivocal answer for that particular question, but we'll raise up the question and you can examine it and think about it for yourself here. So one level of expression that you can look at are the different levels of substantial, moderate, low, inconsequential, or the easiest to interpret, not expressed at all. And the method we use is RNA sequencing. So you capture each messenger RNA, you amplify them a little bit, they get sequenced, they're aligned to the genome, and then you count how many sequences for any particular transcript you have. And this gives you the level of expression. And lately, we've been using this machine called the NovaSeq 6000, which is one of the latest Illumina instruments, and it allows you to look at 2 to 3 billion fragments sequence per run. Each one gives you 150 bases of information. So you can imagine that you can generate an enormous amount of data, or put many replicates into of a particular experiment or time course dose response in any experiment, and get very deep sequencing on 1 piece or 2 piece or 3 different brain regions, whatever you're interested in as you proceed. So the power of this is quite immense. And you know, as I mentioned, it's basically a counting technique for expressed genes. The advantages over microarray are, we know what 0 is. We can quantify low, high, and very highly expressed genes fairly accurately. We can look at splicing. And the example of all of this is shown right here for the opiate receptor in the mouse. So you can see if these are the different exons in color, and you get a lot of reads for exon 1. You can look at these junctional reads. There are fewer of those, but that span the splice between exon 1 and 2, and plenty of reads for exon 3 and 4. So this way you can look at not only the expression but also the splicing, and verify a variety of different hypotheses. As is exon 21 in ion channel X, Y, or Z? How well is that expressed, and what's the level of splicing that might go on? And one example that we examined early on was this particular molecule, gastrin-releasing peptide. So a paper had been written on gastrin-releasing peptide receptor mediates itch sensation in the spinal cord. And I had sequenced, this was in the mouse, I had sequenced the mouse DRG quite deeply, and I kind of knew that there was no GRP in the mouse spinal cord. And I mean, in the mouse dorsal root ganglion. So, what's the story? This is nice looking staining, right? And if I had gotten the staining, I'd be pretty excited about it. But it clearly is related to a cross reaction with substance P and the neuromenin. So if the transcriptomics shows us no expression, you have to be able to account for this sort of staining that you see in the DRG. And then when you look at the sequence, you can see that the carboxy end of besin, a molluscin peptide, neuromenin C, neuromenin B, substance P, very highly expressed. They all contain this methionine amide, which is a strong determinant of immunoreactivity for any kind of any of the antibodies that have been generated. So this was basically a cross reaction. So that kind of lets you realize, well, the power of this is that I can verify what's in the tissue, and should I or should I not examine it and troubleshoot some of your experiments. Now, if we extend it into the spinal cord, the picture's not as clear as it was in the dorsal root ganglion. Here we're looking at gastrin-releasing peptide. It's actually expressed in the human spinal cord, maybe not the greatest level of expression, but it is present and you can measure it. And that's the same across species. And when you look at the this peptide here, this receptor for this peptide, which is theoretically released from the dorsal root ganglion, this is found in PBB. It's not found very much in the spinal cord, but it's quite high in the dorsal root ganglion. This is probably the itch receptors identified by Mishra and Hoon a while back. And it interacts with the, from the dorsal root ganglion. And it interacts in the spinal cord with these NPPB peptide receptor 1, 2, and 3. So this is the main one. And you see that it's expressed in the mouse. It's found in the rat, but it is expressed well in the mouse, well in the rat, but not all that well in the human. So you have to ask the question, well, is 5.79 enough? And to give you an example of like a really highly expressed gene, like neurofilament is expressed at 5,000 RPKMs, whereas here we're talking about 5. But interestingly enough, it is these receptors that interact with neuropeptides are not necessarily expressed to the highest level, yet can still have quite a bit of function. So this requires maybe looking at it in a different way, and to address the question of, is it in the cell that I'm interested in? Is it in the circuit that I'm interested in? And in this case, even if you don't have an antibody, you can use in situ hybridization to figure out where it is. Before we get into in situ hybridization, I'd like to show this section from this is a section from human spinal cord from AnaBios, and we're staining it for CGRP. So we've got a paraffin embedded, formalin fixed, paraffin embedded 6 micron section, immersion fixed at the time of tissue recovery, and we're staining it. Here you see the dorsal horn, the ventral horn down here, very strong reaction for calcitonin gene-related peptide. And the tissue preservation is really quite nice. Here's a higher power view of that same region, and then even higher, you can see all the terminals. dotting and surrounding the cells. Interestingly enough, the cells in the human dorsal spinal cord and in the rat for that matter, but are quite small. And there's a lot of them, but they're quite small in terms of cell body size. So this is an example that using this methodology, we can look at human tissue and get very strong and good looking results. And then we might want to ask the question, well, how can these techniques be used to identify a path to a new analgesic drug? So we can look for pre-existing validation of a target, and the corresponding cell. Some of the questions that you might want to answer are, does pharmacological activation or inhibition produce analgesia? Does human genetic mutational evidence exist? Does an animal model of genetic manipulation, such as a knockout, also verify functional validation? And is the target tractable? And, you know, one easy question about tractability is it a cell surface plasma membrane located ion channel receptor? So that you can get, so a drug can get to it relatively easily. And then we can look at expression in cells and circuits, as I noted before. And is it in the peripheral or central nervous system? So how specific is this to any one system or any one brain region? These are really important questions as one might proceed along the pathway to drug development. I myself am a very firm peripheralist, so I like to look at the dorsal ganglion. I'm venturing into the spinal cord a little bit right here and looking for specificity and things like that. So what's an example of a molecule that meets all of these criteria? Well, the spinal opioid system is one. In 1976, the Action Rudy showed that intrathecal injection of an opioid, intrathecal injection of an opioid produces analgesia. In 1975, it was shown by Kurt and Kuhar that there were opioid receptors in the spinal cord. And now we're about 50 years later looking at this in a more defined fashion. So, you know, pharmacologically, we find that it's there. Genetically, you knock out the mu opioid receptor and it abolishes analgesia. And the circuit, the neurons are present in the dorsal spinal cord. You know, and the next line here is, but wait, aren't we supposed to be getting away from opioids with the opioid crisis? The answer to that is yes. But the cells in the circuit are wired up to produce analgesia. So this is an interesting element. Therefore, other components in these neurons could be used to control the circuit, maybe not directly through opioids, but some other component in the cell. So what we want to look at is a systematic analysis using in situ hybridization and single nucleus sequencing to answer the question, what are the molecular targets for neuronal control? So this shows the mu opiate receptor and a couple of others, the substance P receptor, enkephalin, proenkephalin, and prodynorphin in the spinal cord of a human being. And we show it against the bright field photomicrograph. And you can see a lot of staining here in the dorsal spinal cord. There's some staining in ventral spinal cord and also around the central canal. And this is L3, this is L4, but we've got phenomenally good tissue preservation. You can see some of the intermediate lateral cell column axons proceeding out, all the afferents coming in. But you know, you can't really get an appreciation of what's going on at this magnification. But here we blow it up a little bit more, and you can see in pink the opiate receptor, in orange the substance P receptor, which doesn't come out all that well because it's also overlapping a little bit with dynorphin, and in green proenkephalin. So they're scattered around here. But if we're interested in the circuit, we have to delve into it a little bit more. This shows you a higher power view. And I haven't really taken all of the different channels and analyzed them individually. There's just not enough time for that. But you can see in this high power view, a selection of the complexity of actually what's going on. And that is, we have some proenkephalin and OPRM1 here, proenkephalin alone right there, OPRM1 here. So there's a fair amount of complexity, and you want to delve into this and pull it apart. So if I was interested in what are the components within an opiate receptor expressing neuron that might give me analgesia, I have to really appreciate what's the range of expression in the dorsal horn, and what is the wiring diagram. So this starts to give you an idea of what the range of expression is in the spinal cord. And we can go with this a little bit more in depth too. So we began to look at this with single nucleus sequencing, and I just want to show you a couple of methodological approaches to this. And that is we obtained nuclei from frozen tissue. This is also from AnaBios. The tissue is homogenized, and the sucrose gradient centrifugation allows us to isolate the nuclei. We spin them to the bottom of the tube, and then we start to do fluorescence activated cell sorting using a two-step process. First, we have a double-stranded DNA dye, DRAC5, and that's shown in the first panel here, where debris, which is not very fluorescent, shows up. There's quite a bit of debris, despite the density gradient centrifugation. And we're getting a couple of populations of nuclei that are bright and brighter. And then we also stain this by incubating with NuN. This is an antibody that, here's the real gene name, which codes for a splicing factor found in the nuclear matrix. So the nuclei are loaded with this, and we can add the antibody to the nuclear isolation and sort for these particles, NuN positive particles. And so these are pretty much NuN negative. And we couldn't decide whether this population, this little one here, is that NuN negative or positive, but it looked positive enough. And because we have so much sequencing power, we just isolated all of those and made a library out of this, all three populations that you can see right here, 1, 2, and 3. So we did library construction on 10x chromium, sequencing on the NovaSeq 6000, and analysis of sequence data. So what does the library look like real quickly? We get nice conversion to cDNA, and this shows that it's pretty similar in terms of size, and these are the molecular weight standards. My mouse technique isn't so great. And then when you analyze a particular run from, we did about 8 or so of these, you can see this barcode rank plot shows you really good sequencing, then it drops off. I mean, conversions, then it drops off. And this is an example of a very good library preparation. And how do we, so, you know, the NuN antibody works, but it's not 100% purification, okay? And so, but without it, we don't get any neuronal nuclei. So this just shows you the entire preparation from a particular animal, I mean human, sorry. And if we leave out the NuN antibody, this population down in here is completely absent. So we know that retrieving neuronal nuclei is really important on this particular antibody retrieval step. And here are 6 different preparations. We have about 22 different populations in the spinal cord. This is all of the different ones, and you can see that looking at NuN, that I get a variety of different amounts of nuclei, neuronal nuclei, and we're still working on that. One of the big variables is probably the amount of tissue that you start with. You can't overload the system with tissue. And this just shows you sort of the range. We have endothelia, oligodendrocytes, astrocytes, 4 different populations of neurons, ependymal cells, spinal fibroblasts, and microglia. And let's take a look at the opioid system and some of the other ones here. Here we have NuN, so that shows you the neuronal population. These are excitatory neurons, inhibitory neurons, and Kefalin is found in both populations. Dynorphin is mainly found in the inhibitory population, but also there's this small population that's also coexisting with the excitatory cells. And then the opioid receptors, quite prominent. The kappa receptor is not nearly as well expressed, and the delta receptor, again, is also not nearly as well expressed, but are fairly distinct populations, although there might be some overlap between kappa and mu, et cetera, at the cellular base level. So then when we look at this, well, we have actually a very large population. One might need to start to break this down into, well, should I focus on the opiate receptors in the excitatory population or the opiate receptors in the inhibitory population? Which one might give me better control? And by analyzing the gene expression profiles in these different populations, we might be able to get a better idea of what we can do. So there's still a ways to go till we reach enlightenment, I'd say. In human spinal cord, we see some OPRM1 receptors are an excitatory neuron, another set an inhibitory. In situ shows most are in the dorsal spinal cord, and the combinatorial co-expression matrix is complex. But I'd say working direct in human may save time. On the other hand, we may have to reverse translate some of these findings into an animal model. So I'd like to say thank you to everybody for listening, and thanks to the team in the Department of Perioperative Medicine, Mary Kate Doherty, Matt Sapio, Ali Manalo, Ellen Stedler, Andrew Manis, flow cytometry with Jagan Merrick, and the molecular genomics core of the National Institute of Child Health and Human Development, Steve Kuhn, James Ivan, and Rachel Lazarus. Thank you very much. And also a big thanks to AnaBios and Andre Ghetti.
Thank you, Mike. This is a terrific presentation. Yeah, we're going to reserve the questions for the end after Dr. Jeevakumar's presentation. Now we're going to switch gear a little bit and look at essentially the same preparation, the same spinal cord samples, but with very different readouts, relying primarily on calcium imaging. Vivek, please take it from here.
Thank you, Andre. Thanks for the opportunity to present this data at the webinar today. So I'll be talking about a lot of the functional data that we've been collecting on the human spinal cord tissue. We've been validating this human spinal cord tissue that we receive at AnaBios, and I've been working on this over the last several months. We've been validating it with immunohistochemistry and some imaging assays. Functional assays include calcium imaging and some patch clamp electrophysiology, which is in the early stages of development at this point. But we look at all of that data today as well. So let's get into it.
Vivek, sorry to interrupt.
Yes.
Could you change your display settings to full view? Thanks.
Better? Is that good, Chris?
Yes, thanks. Yes.
Okay. Okay. So we receive these human spinal cords at AnaBios, and we want to get from a human spinal cord that we see here in the image on the left to this. Now, this is an image of a slice that was loaded with the calcium indicator dye, and we do calcium imaging with this, of course, and I'll play this video here. And you can see the calcium transients in these cells, and we see a lot of spontaneous activity in this video. Of course, this video has been sped up to 200 frames per second, but you get the idea. So we want to get from the spinal cord to this, and that's what I'll be talking about today. Okay, so we have proprietary tissue procurement procedures, and we transport the tissue to our lab in a proprietary solution, as we can see here in a container on the image on the far left. So once the spinal cord is received, we do a lot of quality checks on the tissue to determine which area of the tissue that we can make slices and process for slicing and culturing. Once we get the tissue, we take segments from different areas of the spinal cord. In the middle panel, where we see the spinal cord image, sections taken from distal thoracic and lumbar sections of the spinal cord. Now, the spinal cord that we get is typically about 12 to 15 centimeters in length. So we take segments from different areas of the spinal cord, and we do TTC staining with these segments to determine which areas have viable cells and which are good for slicing for culturing experiments. Now, once we determine the location of the spinal cord, then we cut these slices on a vibrotome to 450 microns thickness. And these slices are again stained with TTC. So we know that we have good viable slices. and we can further use those for culturing and functional assays. And we prepare organotypic cultures, and we validate this tissue, like I said, using functional assays, which will include calcium imaging and patch clamp electrophysiology. And we've also been working on some immunohistochemistry experiments with these slices. And of course, we continue to validate our tissue over the course of experiments. So here I have an image of live dead staining done on a couple of slices from 2 different donors. The image on the top, live dead staining done, and we see a higher number of cells that have Ethereum bromide staining. And obviously that tells us that this tissue isn't of that good viable quality. But over time, we've been improving our surgical techniques and tissue processing procedures, and we've been able to get to a point where we can get more viable tissue as well. So that's what we see on the panel on the bottom. More live cells stained with calcine, and we get a higher percentage of viable cells in this tissue. And I've also drawn an outline of the substantia gelatinosa, so everybody can orient yourselves to where we're looking at in the tissue. All right. Okay, I'll start with the calcium imaging assay. So we do imaging on our slices between day in vitro 4 and day in vitro 20. That's the longest we've tested our tissue, and then we've been getting viable tissue up to DIV 20 in culture. We used the calcium indicator dye flow 8 AM for our calcium imaging assay. Excitation at 490 nanometers and emission 520. We used an inverted wide field microscope and acquired data at 2 Hertz. And image analysis is done with drawing ROIs and ImageJ. Most of our analysis is confined to the superficial dorsal horn of the spinal cord. And the readouts from the calcium imaging assay, the del-F over F, we look at the counts per minute or the frequency of these calcium transients as our marker of network activity, and we can then apply various compounds, drugs, to see how this counts per minute or the frequency of the events is modulated by any sort of pharmacological manipulations. And the image on the right, of course, is of a slice that was loaded with the flow 8 AM dye, and which we would use for calcium imaging. Okay, so this graph here shows that we have quite consistent, stable, spontaneous activity over multiple days in culture. The data here is for between days in vitro 4, 5, 6, 8, 14, and up to 20. And these are the different points at which we've been able to do imaging on our organotypic cultures. And we can see that we get a counts per minute on average about 0.5. And it remains stable over long periods of time in culture. So this tells us that if we were to design experiments where we want to look at long-term viability of cultures and do pharmacological manipulations over multiple days in these organotypic cultures, we have the system in place to do that. All right, so getting into the data, here's an example of a slice that was imaged on DIV 20, and this was only imaged in ACSF, the standard ACSF with standard calcium, magnesium in the solution, carpet and bubbled ACSF, and we can see that the slice shows sustained spontaneous activity for up to 35 minutes. Now, it's just an example of a slice that was imaged up to 35 minutes. Typically, our experiments last an hour, hour and a half. So having stable spontaneous activity is important for us over long periods of time. And this tells us that we can design experiments for extended periods of time with our tissue. And I'll also quickly talk about the images that I have on the slide. On the left, we have a raster plot of the calcium transients. The amplitudes were plotted. Red indicates large amplitude, blue smaller amplitudes, and the middle panel shows the raw traces of the del-F over F of these cells. And the right panel, of course, shows us the stability of the spontaneous activity over time. Okay, so next let me get into the imaging and functional validation of the spinal cord tissue that I've been working on. So first, I'll start with some immunohistochemistry work that we've been doing. So in the middle of the slide, we have a pie chart showing the proportions of different interneurons in the rodent spinal cord. We want to be able to create a map similar to that with our human spinal cord tissue as well. So we've been working on quantifying the different types of interneurons in the slices in the dorsal horn, and I have some example images here. So first on the top, calretinine stained cells, low magnification image on the left, couple of cells at 60x magnification on the right. These calretinine expressing interneurons, excitatory cells. I mean, these are known to drive spontaneous activity in the dorsal horn of the spinal cord. So we want to be able to quantify these. And we've also been looking at other inhibitory interneuron types as well, staining with NNOS. PAX2, that's expressed in a very high percentage of these interneuron population in the spinal cord. We want to be able to quantify that as well in the human spinal cord, and galanin, of course. So we're building up this database at this point. Now, related to inhibitory cells, looking at disinhibition in the human spinal cord on the image panel on the right. So combining, our knowledge of the immunohistochemistry data, and then looking at functional assays, when picrotoxin and strychnine are applied, the GABA blocker and glycine blocker, we are able to get disinhibition of the spinal cord. So we can see that there's an increase in spontaneous activity, as shown by the graph on the bottom right. Further validation of this tissue, so we look at excitatory glutamatergic transmission as well. And when CNQX is applied, or CNQX in combination with AP5 to block AMPA receptors and NMDA receptors, we get a reduction in spontaneous activity, of course. So these tell us that we're the counts per minute of the calcium transients is a good measure to be able to validate the human spinal cord tissue. Okay. We've also looked at applying inflammation in the spinal cord. Application of PGE2 and bradykinin in combination also, of course, increases spontaneous activity in the dorsal horn of the spinal cord. So that's shown here in the raster plot, and you can see the raw traces on the top right, and the normalized counts per minute in the bottom right panel, showing that PGE2 and bradykinin show an increase in spontaneous activity. Okay, so as Andre mentioned earlier, Mike Hildebrand and his lab member, and Marie Tedek, they've been serving as advisors for our spinal cord project. So this was a paper that Anne-Marie published a couple of years back, looking at BDNF treatment in the human spinal cord. And so what we see here is that BDNF treatment for about 70 minutes, as she had done in her work, increases NMDA receptor mediated neurotransmission. This was work done in using patch clamp electrophysiology. NMDA receptor EPSEs are increased, and this also happens through GLUN2B subunit dependent mechanism. So we want to be able to see if we can replicate some of this work with our calcium imaging assay. So what we did was we treated our human spinal cord slice for 90 minutes with 100 nanograms per mil of BDNF, and we can clearly see that the spontaneous activity is much higher in the BDNF treated slice. The control bar is an average of our historical data, looking at all of our previous spinal cord slices in vehicle treatment conditions, and comparing to that, we get a much higher increase with median of treatment. So again, it gives us further validation of our tissue in comparison to previously published data. And of course, we are also assessing the role of GLUN2B subunits in this increase in spontaneous activity in the human spinal cord slices, and we hope to have that data out soon as well. Okay, another area that we've been using our human spinal cord slices is in a few safety assays. So quick background on the idea behind this project. So we want to create a ranking of sorts of different molecules that are anti-epileptic drugs or used in the treatment of pain. Now, of course, the pharmaceutical industry relies on rat behavioral readouts to predict CNS effects of drugs. And the Irwin and Rotrod tests are quite commonly used in these tests to look at adverse events. Now, the question is, how predictive are they of the Irwin and Rotrod tests? Are how predictive are they of adverse events that can translate directly to humans? So, commonly reported adverse events such as dizziness, headache, these tests may not be able to predict them as reliably when translating that from experiments done in animals to humans. But with our organotypic cultures, we have a unique opportunity to test these compounds directly on CNS tissue and be able to provide a ranking of molecules based on their efficacy in the CNS tissue in spontaneous activity. So we've been looking at different compounds that are, like I said, either anti-epileptics or used in treatment of pain. So far, we have data from 4 different compounds: carbamazepine, lacosamide, mexiletine, and vixantragine. And I have the free therapeutic plasma concentration range as well in the table, and some of the commonly reported adverse CNS-related effects. And again, the reliability of our spontaneous activity assay in the spinal cord, we can see that here. So the gray bar, of course, is the free therapeutic plasma concentration for each of these drugs. And we can see how different drugs behave in their free therapeutic plasma concentration range. So based on this data, we could make a deduction whether a certain compound is safer or not, in their CNS effects. For example, comparing panels A and D between carbamazepine and vixantragine, in the free therapeutic plasma concentration range, we already have a reduction of about 40% in spontaneous activity. And comparing that with vixantragine, which is a more well-tolerated drug, we can see in the panel D that we don't see that much of a reduction in spontaneous activity within its free therapeutic plasma concentration range. Okay, moving on to patch clamp electrophysiology. I'm an electrophysiologist, as Andre mentioned, and during the introduction section of the webinar. And this is one of the things that I enjoy the most about working with the spinal cord tissue, but obviously this is the most challenging part of this assay as well. We've been able to get to a point where we can start patching neurons in the dorsal horn. Images on the top show the patch pipette where you can orient yourself in the substantia gelatinosa. The 2 images on the left and the middle, and on the right is a 40x image of a cell that was patched, and we can get the spontaneous EPSCs from neurons in the spinal cord in the SG. We can look at both EPSCs and iPSCs, changing the holding potential of the cell. Inward currents in the blue traces in the middle bottom, EPSCs, spontaneous EPSCs, and outward currents in the black traces on the top. where we look at spontaneous iPSCs in these cells. And of course, we also did an experiment where we looked at the effect of inflammation on spontaneous iPSCs in the neuron. And you can clearly see that the frequency and the amplitude of iPSCs is increased after co-application of PGE2 and bradykinin. And this, of course, matches well with the spontaneous activity data that we get in the calcium imaging assay, as well as we saw earlier. Okay, and next, moving on to another aspect of this human spinal cord tissue. That's that this is under development at this point, but quite exciting data. So we want to be able to see if we can reestablish the DRG to spinal cord connections in our slice culture system. There is data, of course, published previously where DRG and dorsal horn neurons make functional connections in the microfluidics chamber and left to grow in culture over multiple days. Functional assays can be performed by stimulating cells in the periphery and doing a readout of calcium imaging activity in the dorsal horn compartment, which contains the cells from the spinal cord. So, of course, microfluidics chamber and dissociated cells, but we want to be able to see if we can accomplish this with our slice culture model as well. So we've been working on that over the last couple of months. And what we see here is our organotypic co-culture system of DRG and human spinal cord. The image on the top left is taken in the DRG section of the culture. So DRG and spinal cord slices are placed in proximity, and they are cultured over, in this example, up to in vitro 14. And then we image them at DIV14, and these slices were stained with fluoromyelin, which is a lipophilic staining, and that allows us to look at the fibers in the spinal cord. So, which you see in the shape of bubbles or round structures in the right panel, and the day before imaging, we inject dextran in the cells in the dorsal horn, so we can see the axons and which direction they've traveled in, how far they have traveled in the spinal cord. So the image on the left again is the DRG. Red is the dextran, and the image on the right is the spinal cord portion of this co-culture system, and we can clearly see axons from the DRG have traveled in the direction of the dorsal horn of the spinal cord. Of course, at this point, we have just these images to look at the co-culture system, but more thorough validation with functional assays is under progress at this point. And of course, a nicer Z-stack confocal image on the bottom here of the organotypic co-cultures.
Thank you, Vivek. So we're now opening to the Q&A session. We already have a few questions that came in. Actually, there's a first question for Dr. Iadarola with regards to the variability in between donors. Do you have data, or do you have a sense from replicas within donors or between donors whether the variability you hinted to in one of the slides is related to the technical challenges of nuclei isolation in the spinal cord preparation, or is more dependent upon donor-to-donor variability?
Thank you for the question. The answer is it's not the donor, it's all in the preparation. And the solution is to not use too much tissue, about 250, well, for our system, we use about 250 milligrams of tissue. When you start to get into 400, you jam up the system with too much debris, et cetera, et cetera.
Great. There's a few questions for Dr. Jeevakumar. So the first one is, was calcium activity detected in non-neuronal cells as well as neuronal cells in the cultures?
Yes. So in the data that I've shown today, we've seen that there's a bar for TTX that I've also shown along with my data. So at the end of our experiments, we always apply TTX for any of the assays that we've done, and that allows us to distinguish between neurons and glia cells. So any of the analysis that I've shown today are cells that had a complete block of spontaneous activity after TTX was applied. So yes, and we do see calcium transients in both neuronal and non-neuronal cells. There are some ways to distinguish between them. The higher frequency of calcium transients, that's typically what we see in the neuronal cells. Glia cells have slower, wider transients, so that way we are able to distinguish between the two different cell types.
Okay. And the second question is, leaving aside the co-cultures between DRG slice and spinal cord slice, if you only have a spinal cord slice, can you stimulate the entry zone and measure postsynaptic responses in the spinal cord using patch clamp or calcium imaging?
Well, electrical stimulation in the spinal cord to look at synaptic transmission should be possible. Now, we've been thinking about also, leaving the dorsal roots on when we make our slice cultures. Haven't had much how do you call it? It's a challenge leaving the roots on, at least when we make these slices, because they're cut at 450 microns. Probably not that feasible to do it, but just doing electrical stimulation in the spinal cord and looking at synaptic transmission, that is quite possible to do.
And another question is, do you leave the meninges on when you slice the spinal cord?
No, we do not leave the meninges on. Removing the meninges is critical because when we're cutting the spinal cord on the vibrotome, it doesn't help to have the meninges around it.
Okay. Another question is, if you want to introduce mutations for gain of function or loss of functions, are there reliable approach for doing this in the spinal cord slice culture?
Yes, there are. Now, the work we've done is, of course, the viability of the cultures. That's something we've been able to maintain over periods of time. So any other manipulations that we may be able to do with the slices, that is definitely doable as well.
So at this point, we have no additional questions. So I will thank the speakers and everyone in the audience who joined us this morning for this presentation. This presentation will also be available offline on our website if anyone wants to re-listen to it. And of course, if you have any questions, further questions, you can reach us on our website or Dr. Iadarola at the NIH. Thank you very much.